Welcome everyone to the second talk of our EHAP 2020-2021 series. The series is entitled Genetics, Evolution, and Human Behavior. And this year, the series is kind of unique because it's being planned and sort of executed entirely by Anthro grad students. So each week we'll have a graduate student host of the speaker who will introduce them and promote the series on social media. So today I'll be introducing Maria Nieves-Colón, who is our speaker. But before I do that, I want to go over some of the technological details. The first is that we'll have a Q&A, and so you can see the icon, it should be at the bottom of your screen. And if you open that, you can type in any questions that you have at any time during the talk. I won't interrupt Maria to ask them, so we'll wait until the end, and we'll have 10 to 15 minutes at that point to go through everything. I'll read your name with your question, so if you want to submit it anonymously, please do that. The talk will also be recorded, and we will publish that hopefully tomorrow on the EHAP Twitter and website and Department of Anthropology website. We also really want to thank Julie Winningham, who is running all of the tech for us and making everything so easy on our end. Thank you so much, Julie. And with that, I am happy to introduce Maria, who is an anthropological geneticist who uses both ancient DNA and modern genomics to examine human population history in the Caribbean and Latin America. Her work uses both paleogenomics and modern genomic datasets to assess impacts of processes such as migration, adaptation, and colonization on human genetic diversity, culture, and health. Maria is currently senior scientist at Claret Bioscience in Santa Cruz, California, and an affiliated researcher with the School of Human Evolution and Social Change at Arizona State University, where she completed her PhD in 2017. Her doctoral research characterized migration, admixture, and genomic ancestry among ancient and modern human groups in Puerto Rico and the Caribbean, in addition to developing laboratory methods to improve DNA recovery from ancient tropical samples. Maria then went on to become a National Science Foundation postdoctoral fellow in the Human Population Genomics Lab at the National Laboratory of Genomics for Biodiversity. Maria's postdoctoral work examined genetic structure and signatures of adaptation in native populations in the Peruvian Andes. Her work has received news coverage from sources including The Atlantic and CBS, and she's done extensive science communication work, including participating in the first Decolonize DNA Twitter conference earlier this year. So we are really honored to have Maria here with us today to speak about an integrative genomics approach to population history in Latin America and the Caribbean. Thank you, Maria.
Oh, thank you, Alex. Can everybody hear me, right? This is my first Zoom presentation, so I hope I can leverage this format to keep it interesting and engaging. It's too bad I couldn't come out to Ann Arbor, but the good thing is hopefully we have people from all over able to join today. So Alex kind of already gave my talk away, what I wanted to speak to you a bit about today is about how my research integrates genomic tools and anthropological approaches to study human population history, and specifically to investigate very big transformative events that occurred in the past and characterize their impact to present day human groups that live in Latin America and the Caribbean. And so I thought I'd start by illustrating how this integrative approach sort of comes to be with an example. So let's consider an event such as this one, the first arrival of Europeans to the Americas in the 15th century. If we only focus on one source of information about the past to study this event, like say for instance, the written historical record or even this painting, we're really just going to focus on the center of this image, on the experiences of European colonizers. And unfortunately, we would be ignoring the experiences of the other protagonists of this event, the indigenous communities who were present at the time of contact and whose lives were completely transformed by this event, and whose descendants continue to live with the ramifications of this event today. But by integrating other ways of knowing about the past, using, for instance, ancient DNA, archaeology, or even oral histories, we can start to reconstruct these the perspectives of these communities and understand a bit more about their experiences. And therefore, we can start to understand the past in a more complete and inclusive way. And this is, I guess, the crux of my research, trying to use genomics as along with other sources of evidence like bioarchaeology or ethnography or even health records to better understand how human populations experience the past and how that past impacts biological variation, culture, and health today. And the area of the world where I'm currently deploying this approach is the Caribbean and Latin America. And I work in this region because it's a part of the world that has a really complex and interesting history of migration, interaction, and transformation. Populations here have experienced everything from adapting to new environments, long-scale migration, experiences of forced displacement, and of course, large-scale admixture. So it's a really excellent case study for investigating how human groups cope with transformative processes. Yet, despite this very interesting history, many populations in this region have been historically marginalized and continue to be underrepresented in human population genomics research. It's working, right? Yes. So an additional focus of my research program is also to drive the inclusion of more diverse populations in human genomics and try to combat our field's current, unfortunate Eurocentric bias, as well as building capacity for future research. So in today's talk, I would like to review 3 examples in which I've been applying an integrative genomics approach to studying the experiences of historically marginalized or minoritized populations from the Caribbean and Latin America. I'd like to start by describing my dissertation research, in which I used paleogenomics to investigate ancient migrations to the island of Puerto Rico and to examine the relationship between pre-contact indigenous peoples and present-day islanders. Secondly, I will pivot a little bit and talk about my postdoctoral work, in which I investigated the impact of population history and health by looking at pregnancy disorders among altitude-adapted Andean families. And I will briefly present, as a sort of third vignette, my current ongoing research examining the genetic diversity of Afro-Caribbean communities. So let's get started by taking you to my homeland, Puerto Rico. So Puerto Rico is located in the Caribbean here. It's at the intersection of the Greater Antilles and the Lesser Antilles in this long chain of islands. And this is a region that's geographically fairly small, but it actually has a really long-standing and interesting history of human habitation. We know from the historical record and from archaeological evidence that the region was first peopled around 5,000 years ago. And these communities had a very dynamic story of migration and interaction between the islands, but also between the continent and the islands. Now, reconstructing these movements has been a central theme of Caribbean archaeology for a very long time, but many questions still remain unanswered or are still being debated. And some of these include how, when, and from where did these populations come from? And what was the scale of interaction between island communities? What we do know for sure is that by the time that Europeans arrived in this region in the 15th century, multiple ethnic communities existed across this region, and one of them in Puerto Rico are the communities that are known to us today as the Taino. Taino is not the name that they used to describe themselves, but it's how we know them today. So it's the name that people use sort of more commonly. Now, indigenous Caribbean communities were also the first Native American groups to experience European colonization. And they were pretty dramatically transformed by this experience. Their population numbers declined quite a lot, and they also came into contact with people from Europe, Africa, Asia, and other parts of the Americas who came to the Caribbean forcibly transported or otherwise. How the indigenous people of the Caribbean and of Puerto Rico specifically responded to this transformation is still a matter of pretty big debate, especially because the colonial documentation record suggests or pretty much indicates, describes, that these communities, they disappeared quite rapidly. They went extinct. And this interpretation of the historical record has led to a very pervasive extinction narrative. The idea that indigenous communities in Puerto Rico and the Caribbean, they just kind of died out, and they therefore contributed very little to the development of later communities and present day people. But this extinction narrative contrasts pretty sharply with what we see in the Caribbean today. There are many communities in the Caribbean who have stories and oral histories of indigenous survival, who have connections, cultural, otherwise, to indigenous groups, and who claim a continuous survival or descendants from these communities. This debate between extinction and survival has really come to a head in recent years because of genetic research. So in Puerto Rico, for instance, it was found about 10 years ago that about 60% of Puerto Ricans have a large component of Native American ancestry in their mitochondrial genome. Now, of course, we also have other ancestries from Europe, from Africa, and other places of the world. But this large component of maternal ancestry from indigenous populations was kind of, well, it really catalyzed a lot of debate. How much of that ancestry corresponds to pre-contact groups, how much of it may have been shaped by later movements was pretty unknown from the study of modern genomes alone. So what I attempted to do with my research was to integrate ancient DNA into these debates. Can we use ancient genomes to reconstruct some of these ancient migrations? And can we also use it to understand or better understand the relationship between ancient pre-contact communities and present-day islanders in Puerto Rico? So to do this work, I collaborated with researchers from Puerto Rico and the US to sample 3 indigenous communities that lived in Puerto Rico before European contact. And these, what we ended up doing is we sampled 124 human skeletal remains from 3 sites, the site of Punta Candelero, Paso del Indio, and Nieves. Here's a picture of Nieves here. All of these sites will be chosen because they were really well studied. We had a lot of information about the archaeological context, and we had radiocarbon dating for most of the remains. And we were able to date them to about 550 to 1300 AD. So this is from about 2000 years ago to maybe 700 years before present. Now, we've mostly focused on sampling teeth and bone, and we did get quite a lot of individuals, but we ran into quite a small problem, as you may imagine, and it's that the conditions that make the Caribbean exceptional for your summer vacation make my job very difficult, because the human skeletal remains were exposed to precipitation, humidity, heat and sunlight, all of the conditions that degrade and rapidly accelerate the loss of DNA after death. So because of this challenge, a large part of our work also included investigating what methods we could use or optimizing methods so that we could isolate the very little DNA that still remained in those samples. And this included looking at different tissues like tooth, calculus, bone, using enrichment approaches, and also lots of next generation sequencing. And so after all of this effort, what we were able to find is that from the 124 skeletal remains that we sampled, we obtained 45 complete mitochondrial genomes, so about a third of our sample. And we also obtained 2 partial autosomal genomes after many rounds of enrichment and sequencing. So with these data, we then began to address our first question. Can we trace the origins of pre-contact Puerto Rican communities through ancient DNA? So to look at this question, we first examined mitochondrial data, and we combined the data from our 3 indigenous communities into one group, which we labeled pre-contact Puerto Rico, and we compared the diversity of their mitochondrial genomes to other populations from around the Americas, both ancient and modern. And this is what I'm showing you here in this multidimensional scaling plot. Each population is represented by a dot. Our pre-contact Puerto Rico population is represented by this red dot here. And the distances between these dots represent genetic distance. So if we zoom into the plot, we can see that our pre-contact Puerto Rico population here clusters closest to these blue dots, which are all populations who today live in the Amazon region. And these include peoples like the Camenza, the Piapocó, and Ticuna, who live in Colombia, Brazil, and Venezuela today. So with the mitochondrial genome data, we see there is some similarity to Amazonian populations. Now, this is great, but mitochondrial DNA is very informative, but it doesn't tell us the full picture. So we also wanted to look at the autosomal genome. So if you'll remember, we were able to recover 2 partial autosomal genomes, and these were both from the site of Paso del Indio. And what we did is we combined these data with another ancient Caribbean genome that was recently published by Schroeder et al. In 2018 from a woman who lived in a cave in the Bahamas, Preacher's Cave. Now, you'll notice that our read depth was much different than what Schroeder et al. Recovered. We were not able to recover as much information as they were able to recover. So our analysis, we couldn't do very extensive modeling analysis. We didn't have enough depth to do that. But what we could do is combine these 3 genomes with a large reference panel of populations from across the Americas and look at admixture and ancestry. And this we did in collaboration with Connie de la Fuente, UChicago. And so what I'm showing you here is an admixture analysis where we're looking at 7 components of ancestry. So we're saying, take all the diversity in these populations and put it into 7 boxes and show me what how that diversity would be distributed. And we've got 3 populations from outside of the Americas as our controls here, few admixed American populations, including modern Puerto Ricans from the 1,000 genomes, and several indigenous populations from the Americas, both ancient and modern. Our ancient Caribbean individuals are here at the end. So let's zoom into them and let's take a look. So what we're seeing here is we have our 2 individuals from Paso del Indio and our individual from Preacher's Cave. And just from a cursory look at their ancestry, we see that they look pretty similar. There seems to be a similar component of autosomal ancestry in these 3 ancient Caribbean genomes. If we compare it to our reference populations, we found that that component is most similar to what we find in other Amazonian populations, like the Biapoco and the Surui. So again, we reiterate a connection to South America and potentially the Amazon region. Now this is actually consistent with archaeological evidence. We know that around 2,500 years ago, there was a major wave of dispersal that brought people from northern South America into the Antilles. These communities, they dramatically transformed the social, cultural, and political landscape of the Caribbean. They brought with them intensified agricultural technology, very elaborate ceramic material culture. And because of this, we think that we call them sort of the first representatives of the ceramic age in the region. Not that there wasn't ceramic before, just not ceramic that looked like that. So this is great. Our ancient DNA evidence recapitulates what we thought we knew from archaeology. But that's not the full picture, because the data that we have does suggest there's a connection to South America, but it doesn't mean there isn't a connection to other parts of the world. So we cannot exclude the possibility that there were other movements as well that we cannot detect with the data set that we have, or that we would be able to detect if we had more genomic data. So the second question that we wanted to address with this data set was, is there any genetic continuity or is there any genetic link between these ancient indigenous populations and present-day Caribbean islanders, like Puerto Ricans, for instance? So to explore this question, we went back to the mitochondrial data. We have a lot more resolution there, a lot more genomes. And we compared the mitochondrial diversity of this ancient sample to a large data set of about 1,000 mitochondrial genomes from across the Americas, present-day populations in the Americas. And to our surprise, we found that the only place that where we had the exact same haplotypes was in Puerto Rico. And that was pretty surprising to us because we do know that there was a large population decline of indigenous peoples on the island. We know that there was a genocide and there was a loss of genetic diversity. So it's pretty surprising to find that these lineages, they're still there despite all of this change. So of course, we wanted to look at these lineages in more detail, and this is what we did next. So here what I'm showing you is a very stylized bar chart representation of mitochondrial haplogroup distributions. We have ancient Puerto Rico here on the left, and our present-day Puerto Ricans on the right. Now these are just Puerto Ricans who have Native American mitochondrial ancestry. We have all their ancestries on the island, but for this analysis, I'm only including those individuals. Some of these individuals may have other ancestries in their autosomal genome. And I'm showing the different haplogroups in colors. So we've got green for haplogroup A2, C1 in the blue, and D1 in the yellow. And what I want to call your attention to here is that there is a similarly high distribution in these 2 populations of haplogroup A2 and C1. This pattern is a pattern that we see across the Caribbean today in many different populations. And that suggests that it's actually quite an ancient pattern and that it's persisted over time despite the transformations brought by contact. Now, if we sort of take a more granular view and we look within each of these haplogroups at sub-haplogroup diversity, we can also see some interesting patterns. Again, A2 is in the green. The different shades of green represent different sublineages. Same for C1 in the blue. It's moving, yes. And then D1. And what we see is that there were 3 sub-haplogroup lineages that persist over time between ancient indigenous Puerto Ricans and present-day Puerto Ricans. And I'm going to highlight a few here. This first one, lineage C1b2, highlighted with the blocks, is actually the most common lineage in the past and in the present. It represents about 33% of our data over time, always, suggesting that this lineage had a really big role and potentially it was part of a founder effect that made it so frequent that it's still the most frequent lineage today. And what's really cool about this is that we know from previous research that this lineage arose in South America about 2,000 years ago. So it potentially could be a hallmark lineage that arrived with these ceramic age movements that came in. There are 2 other lineages that I also want to point out, A2Z and A2 plus 16218. These lineages are very exciting because we don't find them anywhere else. We've only ever seen them in the Caribbean so far. Who knows, next year. But that suggests that these lineages could be a local development. It could have evolved in the Caribbean and they're still present in the populations that live there today. Now, I also want to point out there are many other sublineages here that we cannot trace to anywhere. We don't know where they came from. We haven't seen them in present day populations. We see also some discontinuities, some lineages that were present in the past that are not present today, such as this one, and vice versa. So there are still some changes that occurred in these genetic pools and of course in these cultures as well because of contact that we can see in the genetic data. Now, I want to mention briefly that since my work came out, there has been kind of an explosion in Caribbean paleogenomics. And in just those last few months, the number of Caribbean paleogenomes has increased to about 300 or more because of these 2 studies by Najili et al. And Fernandez et al. And their work has really expanded upon our findings. They also found that there was some continuity over time, but they have also been able to look at other more ancient movements in the Caribbean and have found that there are multiple, or there's evidence for multiple dispersals, some of which we know are from South America and some of which we have absolutely no idea where they came from. So many, many more questions arise when we lose ancient DNA to look at the past. We answer some, we have some more questions. That's how it goes. Keeps us all employed. So let's summarize what we found. So overall, we found that there were 45, we were able to recover 45 complete mitochondrial genomes from 124 ancient individuals from Puerto Rico. And this was kind of a challenge, but it was a really fun one, and we were able to successfully recover some tropical ancient DNA in this context. We found that the ancient population of Puerto Rico had a very strong link to South America, but we cannot exclude other potential origins, of course. However, these findings support insights that we've seen from other lines of evidence, including archaeology, modern genomics, and biodistance studies, all of which indicate that South America is a primary source region for pre-contact Caribbean peoples. We also saw there was similarity in autosomal ancestry components across different Caribbean populations, a finding that Najili and Fernandez et al. Also saw, suggesting, of course, that indigenous Caribbean groups had a shared ancestry and may have diversified after kind of coming from at least one region, maybe more. And lastly, we also found there was some genetic continuity in mitochondrial genome diversity over time. This indicates that despite the large scale of post-contact demographic shifts, we do see some connections between present day and ancient people in genetic connections. We knew there were cultural connections, but now we also have some genetic evidence for that. And this last point is, I think, quite important because at least in Puerto Rico, and I know in other islands of the Caribbean as well, the indigenous component of our culture and our identities is really important. It's actually one of the central parts of our sort of national and ethnic identity. Indigenous artwork and symbols are used today as symbols of political empowerment, of cultural belonging, and they're pretty essential as part of what defines Puerto Ricanness or being Boricua, as we would say it ourselves. But this indigenous pride coexists with an indigenous erasure, which is part of this extinction narrative that I mentioned earlier. And the best example of this is my 10th grade history textbook, which says Puerto Rico 500 years of history, not 5,000 or 4,000, 500 years. It all started when Juan Jose León arrived. And this contributes to an erasure of not just our indigenous ancestors, but also our Afro-descendant and African ancestors as well, who are also kind of given the short stick on this in this book. Now, indigenous communities and indigenous descent communities in the Caribbean have fought this extinction narrative for a very long time. And especially in recent years, because they've been gaining a little bit more political power and recognition, they've been saying this story is more complicated than just these people disappeared and we completely started anew. And genetic data, including my work and the work of others, as well as the press coverage of that work, is really changing this debate and how people in the academic community, as well as the public, relate to their past and understand the indigenous component of our story. So much so that today there are many efforts for Caribbean root seeking, which are formed by the use of genetic technology. So this big role for genetic data and the data that we put out there is not going to change. It's in fact, it's going to increase. And this is something that I'm really interested in exploring in the future, how people's conceptions of the past in this region are changing because of the introduction of direct-to-consumer testing, as well as my own research that I'm putting out there. So returning to the main themes of my talk, the ancient Puerto Rico study is a really good example of how we can use ancient DNA to reconstruct population history and understand the implications of that history for present-day culture, as well as identity, and enrich archaeological research. But now I would like to switch gears a little bit and tell you about the second component in my research program, which is how we can use population history, or how we can understand how population history impacts human health. And I will illustrate that by talking to you a bit about my postdoctoral work, in which I investigated how adaptation and admixture influence reproductive health among altitude-adapted families in Peru, in the Peruvian Andes. And specifically, this work focuses on a disorder of pregnancy called preeclampsia. So preeclampsia is a pregnancy complication which is caused by abnormal placental development. It can result in abnormally high blood pressure in the mother, and serious complications from it include multiple organ failure by the mother, fetal restriction, growth fetal restriction, premature birth, and even maternal death. In fact, worldwide, preeclampsia causes nearly 40% of all premature births and is associated in at least 10% of all maternal deaths. And despite the fact that it's so dangerous, currently there are no preventive treatments or early diagnostics. The only thing that they can do is induce the mother to give birth, which is why it's involved in so much premature death. And despite the fact that we have known since the 19th century that this disorder is heritable, it's heterogeneous as well. So there are both genetic and environmental causes. And that means that it's very difficult to isolate one thing that can cause it, and therefore to identify effective treatments. Now, all women around the world are at risk of developing preeclampsia with pregnancy, but this risk is higher in developing countries and among indigenous communities who may lack access to adequate healthcare. Additionally, populations that live at altitude have 2 to 3 times more risk. So this condition is actually really common in places like Colorado or in Bolivia or Peru, the Andean region. In Peru specifically, preeclampsia prevalence is 3 times the worldwide average. It's the second leading cause of maternal death. Now you may be wondering, well, wait a minute, don't Peruvian, especially Andean populations, have altitude adaptation? Well, we've known for some time now, for a long time now, that native Andean peoples have adaptations that are physiological and genetic for the extreme conditions of high altitude hypoxia. So how could that be impacting risk for developing pregnancy disorders that are exacerbated at altitude? Now, there's been some research into this topic, and it suggests that perhaps some Andean ancestry may play a role in susceptibility and risk to these types of disorders. Perhaps some of that adaptation may actually provide protective variants that reduce risk. Andean populations are sort of this really cool natural experiment. I kind of hesitate to call them that, but because they have a unique adaptation they have high disease burden, and they have a lot of selective pressure to sort of deal with that issue. And because of that, we thought it would be really interesting to study this population and see if we can investigate some of the genetic basis for preeclampsia in an altitude adapted cohort. Now, in order to do this, I joined a very large collaboration that was studying preeclampsia in the Andes. And this was a group of clinicians and scientists from Peru, Mexico, and the United States. And they had done a longitudinal sampling of preeclamptic families at the regional hospital of Puno, Peru. This is a city that's about 13,000 feet above sea level, 5,000 meters for the metric system. And what was really cool about this sampling is that we focused on trios. We had mother, father, and child, well, actually umbilical cord, and we collected cord blood for the offspring, blood from the arm for mom and dad. For the mom, we also had some placental data, and we had medical records. This was data for over, I think, 400 trios. And a big part of this project was also that we wanted to collaborate equally with our investigating collaborators in Peru. We wanted this to be a bidirectional training experience where I would learn something and they would learn something too. So in 2018, I traveled to Lima to work with our collaborators at Universidad Peruana Cayetano Heredia, Carla Gallo and her team. And we brought consumables and equipment and we did some training, and then they trained me. I had never worked with whole genome, whole blood before. This was first experience. And they're just badass. They can pump out like 100 extractions a day. And together we managed to extract DNA from over 1,000 samples, of which we selected 947 for SNP genotyping. We generated data for about 800,000 positions across the genome using the Affymetrix Axiom Lat array, which is specifically designed for Latino populations. And these data were then analyzed in collaboration with an all-star women in science team at the Stanford Department of Biomedical Data Science group. And this included graduate student Kayla Vadillo, who's my co-author on this paper that we're preparing now, and Janet Washik, who is now an assistant professor at Johns Hopkins. And so the first question we wanted to address with these data is, can we detect any ancestry-related susceptibility to preeclampsia? Could there be any differences in, for instance, ancestry proportions or admixture patterns between case families and control families? And we thought this could be an interesting question because of the evolutionary history of Andean populations. We thought, well, perhaps controls will have more Andean or Native American ancestry than cases. And maybe if that's so, we can look at these regions of the genome that are enriched for Native American ancestry to find variants that are involved in this condition. So that was great in theory, but then reality set in, and we found that there were no significant differences in ancestry proportions between case and control families. And so what I'm showing you here are local ancestry plots. This is very similar to what you would get if you did like a 23andMe test. So we have each of our chromosomes here, and then different ancestries are colored. We have green for Native American ancestry, and blue for European ancestry. And you can see in these two individuals, it's a pretty similar pattern, mostly Native American ancestry. These individuals are pretty representative of our two case and control cohorts. So there were no significant differences. If, in fact, ancestry does play a role, we cannot detect it with the methods that we're using here, and we would need to rethink that. So because of this, we decided to pivot our study and focus instead on, can we identify any candidate genes that may be associated with preeclampsia in this population? Now, normally we could do like a genome-wide association test to do this. But in this case, we decided to leverage the family trio structure of our sampling to find association. And we did this through a trio-based transmission disequilibrium test, so TDT. And what this does is it allows us to identify linkage between a phenotypic trait, in this case preeclampsia, and a genetic marker. And we're trying to identify those alleles that are being passed on more often than we would expect under Mendelian rules of inheritance, which we can discuss whether or not are appropriate models to use, from parents to offspring in the affected population. And we have a related follow-up study, which is a TDT-POO test, and that allows us to find parent of origin effects. So do we see this transmission happening more from the mother or from the father? So what did we find? So the TDT identified an association between preeclampsia and several factors, clotting factor genes on chromosome 13. These genes, the SNPs that we found that were our top hits, are in linkage disequilibrium from each other, and they're involved with the clotting cascade. So these include genes like ProZ, F7, and F10. And all of these are involved in the processes that control our blood clotting capacities. You can see here the TDT result. And specifically, our top hit was a SNP called RS5960, which you can see here in this locus zoom plot, which was also the top hit of our TDT-poo test. And we found that, in fact, it had a strong paternal origin effect, not maternal, which is kind of interesting. So basically, this variant is inherited more often than you would expect by chance from the father in affected family trios. Now, what these findings suggest is that irregular coagulation may be a very important factor underlying preeclampsia in a high-altitude adapted population. And in fact, previous research has found that placentas from preeclamptic pregnancies often present irregular blood clot patterns. And this, of course, would affect the blood flow between the uterus and the placenta. So at least in mice models, we know that hypoxia exacerbates thromboses, it exacerbates irregular clotting. So it would kind of make sense that genes involved in the clotting cascade, such as ProZ, would be potential targets that we should look at for understanding the genetic basis of preeclampsia. Another thing that we know from previous research is that ProZ protein expression levels are associated with thrombotic disorders in general. So this, this genetic marker may be important in preeclampsia in this population, but we should also potentially explore it to look at preeclampsia in other groups. The other kind of cool finding that we have is that we found that there was a role for the father, or for the transmission of genetic markers from the father in this condition. Now, our study is actually one of the few to include trio family members in genetic cessation tests for reproductive disorders, because more often than not, these studies include the mother and the offspring. So we don't actually know a lot about the father, even though we know that most pregnancy complications are the result of genome, or have a factor for genome interaction between all the individuals involved, so 3 individuals. And this is actually really interesting to me, because from evolutionary theory, we know that maternal fetal conflict is potentially one of the factors that may influence the persistence of human preeclampsia. But we don't actually know what the role of the father would be. Is there any paternal offspring conflict with the father? We have no idea. So finding this kind of parent of origin effect from the father was kind of opened that question for us. Is this something that we could explore further in the future? Now, in addition to understanding evolutionary basis of this disorder, one of the broader aims of our project was also to inform potential health and screening interventions so that we could identify preeclampsia, or early detect preeclampsia in at-risk women. So given that we found ProZ was a potential candidate loci, and we know that ProZ protein levels are involved in other disorders that are involved with coagulation and pregnancy, we decided to test whether ProZ protein levels could be used to identify at-risk women. So my collaborator, Kayla, did this work. She tested protein levels in a subset of our cohort using ELISA, but unfortunately, we did not see significant differences in protein levels between cases and controls, or between cases that had more mild or more severe forms of the disease. Now, this assay was impacted a little bit by a kind of a small sample size. We're also relying on medical categories of diagnosis that occur right at the time of birth. So maybe some of those variables are not ideal. But in general, we need to do a lot more work before we can translate our findings into potential screening approaches in the future. So to conclude, in this research, I worked with a large team of collaborators to investigate the genetic basis of preeclampsia in a high altitude adapted family cohort from the Peruvian Andes. We found that ancestry and recent admixture do not seem to be playing a huge role in impacting preeclampsia risk in this population. If, in fact, adaptation to high altitudes is conferring any protective variation or reducing risk, we were not able to detect that here, and it may be that we may need to do whole genome sequencing or look at exomes or even RNA-seq to understand this further. We also identified several candidate-associated loci, the most salient of which was this ProZ gene. And that suggests that blood clotting factor genes may impact preeclampsia in this population, especially due to their potential role in coagulation irregularity. We found that because of our trio sampling design, we were able to identify a strong paternal inheritance effect with associated alleles underlying potential interaction between parent and offspring. But of course, this story is far from over. These associations may be very specific to this population. We will need further research with larger sample size, more diverse cohorts to confirm these findings and how applicable they may be to other populations around the world. Functional studies would also in the future help us see how these markers may be actually affecting biological pathways. But nonetheless, this study is kind of a good illustration of how we can use population history to guide our exploration for functional and medically important variants. And of course, especially if we are thinking about developing precision or clinical genomics initiatives that can help these communities, really need to get a better grasp of their underlying structure and how that may be affecting their health. And so the last point I want to make is that this is a great example of why we need to include diverse populations when we want to study the genetic basis of heterogeneous human disorders, especially if we're trying to find genetic markers that may be influencing complex traits, for instance. And since we know that genetic risk and association transferability is really not great, like you can't just take an association in one population and apply it to another, we really do need to get a more full spectrum of human variation. So overall, this work has combined an anthropological perspective with methods from population genomics and genetic epidemiology to examine the impact of population history and health. But in the brief time that I have left, I want to take you back to the Caribbean to tell you about my current ongoing research, which I'm very excited about, in which I go back to examining the past, but this time looking at high-density modern genomes in order to investigate the Afro-diasporic experience in the Caribbean. So the Caribbean was the first region of the Americas to receive the forced migration of African peoples as part of the Atlantic slave trade, which began in this region in the 16th century. It's estimated that over 7 million people were forcibly transported from Africa to the Caribbean during this time. And although all islands in the Caribbean were impacted by this process, the nature and the volume of the trade was different across the region, and the experiences of the Afro-descendant communities that formed on each island are also distinctive. These differences underlie the diversity and heterogeneity of Afro-Caribbean cultural expressions that we see in this region today. And they also likely shape complex biohistories and a very diverse Afro-Caribbean genomic landscape. But unfortunately, that landscape remains quite understudied, even though this is changing now. So part of my work here, what we're trying to do is to use genomics to get a more complete picture of the Afro-Caribbean experience and integrate that with the historical and ethnographic record, so we can understand a little bit more about how these communities came to be and how different migrations that occurred from the colonial period into today have impacted them. Now, for this work, I'm collaborating with Jada Torres from Vanderbilt University. She had previously sampled 5 Afro-Caribbean communities in the Lesser Antilles, including communities from St. Kitts, St. Lucia, St. Vincent, Grenada, and Trinidad. And so we genotyped 72 individuals in total from these communities using the Illumina Mega X array, which is an array to look at diverse and admixed populations. And the first thing we examined was, can we see differences in genetic structure between these groups? Do they have different patterns of admixture and ancestry? And the short answer from our pilot data is yes, indeed they do. They are not all the same. So here I'm showing you, again, another admixture plot. And what we're seeing here is an analysis estimating 5 ancestral components. We have reference populations here in Africa in the red, Europe in the purple, Native American populations in the blue, orange and green for South and East Asia, respectively. And our Afro-Caribbean populations here, you can see them zoomed in here. And we see kind of patterns that we expected. So we see a lot of African ancestry in these groups. This is what we expected. We also see that at least 10% European ancestry is present in all of these communities, a very common pattern for Afro-descendant groups in the Americas. But we also see some differences with the rest of the Caribbean. So in contrast to places like Puerto Rico, for instance, we have a much smaller proportion of Native American ancestry. And we have a comparatively larger proportion of East and South Asian ancestry, especially for Afro-Caribbean communities in Trinidad. They have a very complex admixture profile, actually. About 5 different continental groups are contributing to their genetic ancestry. And this makes a lot of sense when we go back to the historical record. So we know that the Caribbean, especially the former British colonies, became a center for Asian migration from places like India, China, Malaysia. After the emancipation of slaves, the British Crown brought these people as indentured servants to work in the plantation economies. And so what we're seeing with our genetic data is that these migrants who came through this pretty much forced labor movement to the Caribbean are interacting clearly with Afro-descendant communities and in some way shaping the formation of these groups to who they are today. And I just want to mention that there are also Indian diasporic communities in places like Trinidad, even to this day, that are fairly large and also contributing to sort of the cultural diversity of these islands. Now, the second question that we're looking at with these data is, can we trace subcontinental origins? Can we look at where within Africa, Europe, or Asia the ancestors of present-day Afro-Caribbeans come from? And we're doing this by leveraging a technique called ancestry-specific PCA. So here I'm showing a local ancestry plot like we saw earlier of an individual from Trinidad who has a lot of admixture from different populations. With ancestry-specific PCA, what we can do is we can pull out those haplotypes that map to, say, Africa, for instance, which here are noted in the red, and then analyze those haplotypes in isolation from the rest of the genome by comparing them to a reference panel of present-day African populations. And that gives us a better sense of where within Africa the ancestors of present-day communities in the Caribbean may have come from. So this is what we did here. I'm showing you basically a PCA plot where we have our Afro-Caribbean populations in the black, and they're down here in the plot. And they're clustering with reference populations from an area of Africa known as Lower Guinea, which is represented here in the orange here. Now, this area includes present-day nations of Ghana, Ivory Coast, Benin, and Togo. And during the slave trade period, this area was called, variably depending on the time period, the Ivory Coast, the Gold Coast, or the Slave Coast. And if we go back to the historical record, we see that this region exported about 2 to 3 million people to the former British colonies during the slave trade. So it makes a lot of sense that we're seeing this connection. Now, this analysis was done before 23andMe published their huge study of Afro-descendant populations. So we want to go back to the table and add in more reference data so we can enrich this and see if we can get a little bit more resolution. And that's part of what we're going to do next. We're also looking at tracing other subcontinental ancestries. So where within Europe or Asia, we want to look at sex bias, whether we can see different mating patterns. And I want to highlight the work of my current student, Daniela Orozco, who's defending her thesis on Thursday. She's going to be amazing. And she has been looking at dating these migrations, trying to estimate when these people came and how this shaped sort of the present day genome of these populations. So to sum up, this is a project that is currently ongoing. I'm really interested in continuing to expand it in the future because it addresses the very large impact of migrations driven by European colonization, the slave trade, and other forced labor movements like the indenture trade system on the genetic diversity and the cultural experience of Afro-Caribbean communities. So understanding how these communities came to be in more detail can help us also to restore some of these links that may have been destroyed through experiences of forced migration and servitude. So overall, in the past 40 minutes, I hope I've convinced you that by using an integrative approach to genomic anthropological research, it is possible to investigate the impact of transformative processes like migration, colonization, adaptation, and how they have impacted human biological diversity. This integrative approach can also help us to craft more complete and more inclusive reconstructions of the human experience as we are able to address stories of communities that have been historically underrepresented through a new lens, and hopefully, if we do it equitably and ethically, ensure their participation in future research and start to build the blocks for international collaboration that is fair and equitable. By further combining genomics with other lines of evidence, we can then investigate how population history is shaping or has shaped the biological variation, culture, and health of human groups, and we can understand the past and how the past affects the present with a more multidisciplinary, holistic, and inclusive lens. And with that, I will end, and I will happily take your questions. And I just want to plug for myself and say that starting in January, I'll be moving to the University of Minnesota to start a new molecular anthro group there. So interested students, please email me. It's cold. I think we'll be fine. I'm not sure, but the university is on the banks of the river. It's very beautiful. So you can come be cold with me together if you want. So yeah, should I end the slideshow, Alex?
You can keep it if
Okay.
you want. Other people might have questions about slides, but thank you so much, Maria. That was awesome. Just a reminder to everyone that you can go to the Q&A button and type in any questions that you have, and I'll read them out for you. The first question that we have is from Beverly. Did you study any imprinted loci in your work on preeclampsia?
That's a really good question. Thank you for asking that. I wanted to, but the techniques that we used couldn't really assess for that. So that's something we want to do in the future. My collaborator Kayla did some capture sequencing, which I didn't talk about in the talk, of the regions surrounding the loci that we found, the sort of candidate genes. And we unfortunately didn't find a lot that the genotyping didn't tell us. So we think we just don't have enough resolution to answer that with the data that we have right now. But it's a really good follow-up. And from Karen, is there anyone specifically working with Haitian DNA ancestry data? I have the vague idea that someone in the Genographic project is looking at Haitian populations, but I may be off the charts on that. But I think there is a study there, maybe, yeah. It's definitely understudied for sure. This next question from Christina. You mentioned that using Hardy-Weinberg equilibrium as a baseline might not be the best method. Can you expand on that? Oh, Christina, hi Christina. Do you mean this for the TDT test? When we did the TDT genome-wide association test? I think I can see the Q&A. If that's what you're referring to, what I mentioned was that the test assumes there's Mendelian inheritance rules governing, oh, hi Christina, that Mendelian inheritance is kind of the assumption, right? We're assuming that under Mendelian inheritance, there should be equal chance that X marker would be passed on from mother and X markers would be passed on from father, but the test looks for deviations from that assumption. Of course, that's kind of an assumption, of course, because if those regions of the genome are not actually corresponding to Mendelian inheritance, then we're stuck. But that's kind of the only model that we have right now to test it, or with the data that we have, yeah. Follow-up studies are important. I have a question, sort of, I don't know too much about haplotypes, but what is your next research question or step when you see these like unknown sub-haplo groups that are involved in these dispersals? Like what do you ask next? Yeah, you know, actually a lot of the ancient lineages that we saw in Puerto Rico have not been found anywhere else, like ever. Like we just don't know where they came from, and they weren't listed in Phylotree. Not all of them, but some of them. This is not uncommon for the Americas. Other researchers have found this pattern as well, and we see it just because there's a discontinuity between ancient and present day genetic diversity. So my thought, what I would like to do in the future is to potentially, if there's enough DNA preservation and we have enough extract that we don't have to go back and sample the bone, generate more autosomal data from those individuals and see if we see other patterns that are distinctive. The research that I've done so far has looked at within an island, but I would also like to see between other islands, and now that we have more genomic data, this may be possible, and we can investigate sort of more about the cultural context of these communities and how that relates to their genetic diversity. Yeah. Cool. Okay, from Sharmi, this was a really fascinating talk. Thank you so much, Maria. I was wondering if you could elaborate on the impact of your research and other genetic ancestry research on the perception of indigenous identity in Puerto Rico, especially related to the typical pattern of erasure of ethnic identity that we see due to colonization.
What direction do you anticipate your research taking in the future, and what is the future of this integrative genomic approach?
Yeah, so I've been thinking about this a lot, especially because of I showed a slide about how root seeking has really taken off in the Caribbean, especially since my study and the studies of others were published. So I think that this is a big question: is how are people now understanding their relationship to the past? And I guess I'll answer with an anecdote. So I went back to Puerto Rico in 2018 to present the results of my work, and I just talked to anyone that would listen to be like, hey, guys, I did this thing, and it tells us something about us. And people came to my talk, and some of them identified as indigenous or indigenous descent, and some did not. And the most common question I got was, but I did a 23andMe test, and I have European ancestry, so how can I possibly be indigenous or have indigenous descent? And so invariably, I always had to explain the difference between sort of cultural, a cultural and a genetic component, that, you know, they're related, but they're not the same. And so I don't know how this is going to play out when everybody has a 23andMe test and they're uploaded all this data to GEDmatch. And I hope that it won't result in sort of exclusionary categories, but who knows? I think it's going to be a question for the future. And I hope that by integrating genetic data as well as ethnographic study, we can get a better sense of how people's perceptions of indigeneity and identity are changing, because they are changing due to this work. That was a long answer.
Yeah, that's good. Okay, I think we have time for a few more questions. This one is from Andy. Thanks for the great talk. The Africa ASPCA plot you showed looked like no PCA plot I have ever seen. There seemed to be a lot more structure in the PCA plot than I'm used to seeing. Is this typical of ASPCA approaches? Can you tell us a bit more about the ancestry-specific component of the PCA?
Yeah, so like I said, basically what we're doing is we're running a local ancestry assessment tool. We're taking the genome of the admixed person. Say we took myself, we would find there's a 3-way admixture pattern, and then we would sort of blank out those haplotypes that we're not interested in, and we would take out only those that have African ancestry and then plot them with a bunch of reference populations. But of course, that analysis can be influenced by many factors. One is that it depends on the amount, how much African ancestry this person has, or whichever ancestry we're looking at. There's a cutoff of which we just can't say more. How good is the reference data set? Like I mentioned, the analysis, we did it before the really big 23andMe study, so we have a lot less resolution between populations. And also how good our merging of the data is with existing data sets. So if data sets are done on many different arrays, that means that we have a lot less SNPs to work with, which means, of course, that any differences we might not see them, or they might get exacerbated depending on how bad or good the data combination algorithm is. Yeah.
Awesome. Okay. I know it's 5, but we can do one more question.
I'm happy to hang out.
From Frida. That was a super interesting talk. Thank you. Are you interested in looking at health-related traits in the African descendant populations in the Caribbean?
Yeah, I would love that. With the data set that we have now, we don't have consent from the people to do that. We can only look at population history, but it would be awesome to go back and, you know, re-consent or do it in a new sampling to look at these issues. But, you know, first we got to get a really good handle on the underlying diversity. And so that's kind of where we're going first. But yeah, definitely.
Okay, last one from Andrew. Can archaeological evidence from Puerto Rico point towards the provenience of the 2 ancestral lineages you identified as being unknown?
Maybe. Unfortunately, the 2 individuals that had those lineages, or actually there was about 3 of them, were from the same site, so we don't have a huge difference in the context for those 2 individuals. We need more data. I know that's always the answer, but we do need more data from the island to look at. We don't even know if there are different patterns of diversity within sites. We have 3 sites, right, that represent the whole island. It'd be great if we had more. We also need more data from more ancient sites, because all of our data are around 2,000 years ago, but there's like a 3,000-year-old gap there that would be cool to explore to better understand that, because we'd have that context. But actually, I just want to briefly mention that there's some ethnographic evidence that some of these lineages may be present at higher frequencies in indigenous descent communities that live like in the mountains in Puerto Rico. There was a previous study that sampled specifically that population, and they found some of those lineages there. So maybe.
To follow up on that, if I know you're not an archaeologist, but if you were an archaeologist, what project would you like design to get like your ideal data to answer that question? I know that's a big question, but.
Well, I'm not an archaeologist, but fortunately I have wonderful archaeological colleagues, and I did do a little bit of field work when I was an undergrad, and so that has created sort of a nice conversation or dialogue with colleagues on the island. And if I had my way, I would work at a site that also has a present day community living there, so that we could integrate not just understandings of the past, but of the links between that past and present day people. And if we could integrate their perspectives into designing our hypothesis, that would be amazing. And I have an idea to do that at one site. It's called Los Indios, which is a similar time period to the ones I showed, long-term habitation site. So I'm hoping that in the future, we can look for strategies to do that and integrate researchers from the University of Puerto Rico, which is where I studied, to be able to do this.
Awesome. Okay, we'll take this last question from Catalina. Hi, thanks for this amazing talk. Regarding your work on preeclampsia in Puno, do you think it may be possible that other genetic variation or even physiological adaptation related to the high altitude adaptation, which is known to affect blood pressure, could be affecting some of the phenotypic health related outcomes?
Yeah, absolutely. And I think that maybe the disadvantage of our approach is that we're limited to just those variants that are present on the array. So if there's other variants that we just didn't have on the array, we just wouldn't have been able to see them. And I think it would be really cool to perhaps do a follow-up study where we could maybe capture some regions that we know are involved in altitude adaptation and a few regulatory regions that are involved in physiology and blood pressure and coagulation to better understand this issue. So Catalina, if you want to hit me up, we can, we can write a grant. So yeah, awesome. Thanks everybody.
Yeah, thank you so much, Maria, for this awesome talk. We all really appreciate it. And I want to remind everyone that we will be back here at the same time next week for a talk by Keolu Fox. So thank you everyone for attending as well.
Thank you for inviting me, Alex, and thank you all. Hope I can see you in person soon. Bye, everyone.